Now let's look at meiosis in a little bit more depth. I gave you an overview previously. So now keep in mind that these are taking place in the sexual cells, and whether forming gametes or they're forming egg cells. Remember that the beginning cell is going to be a diploid cell, and the diploid cell would then this is the 2n, which is called as the diploid cell, and the diploid cell is going to form haploid cells due to due to meiosis and the process that we will learn here. So let's look at a little bit more depth on the crossing over and see how this different stages takes place. Meiosis I, which is the first part of this reproduction, is actually also called as the reduction division. It's reduction division because it's reducing the chromosomal number. The chromosomal number, which is the diploid number, is reduced to the haploid number. Also important at this stage to remember is the diploid cell is 1. So there is one diploid cell, but it results in 2 cells, and each of them are haploid. The second most important thing that takes place is crossing over. And you can see over here, the crossing over is shown nicely in this image over here. You can see how the homologous chromosomes are twisted, and they're having synapses and breaking pieces from one chromatid to the other. And the last part of meiosis I is called as independent assortment, which we will see in which the chromosome will be separated. So here, after crossing over has taken place, and just before metaphase, the chromosome is going to look like this. So you can see over here the chromosome is actually, it seems like the chromosome is not completely 2 red segments over here. You can see there's a blue segment over here, and there's a red segment over here. So these 2 pieces were swapped. Again, on the shorter chromosome on the top, you can see, let me get my green pen, it will be easier to see. You can see over here and over here, you can see the segments that have been swapped. So this would be, this would all take place under prophase. So prophase 1 is a very long, lengthy stage under meiosis, because a lot of changes are taking place within meiosis, and those are different stages that are unlike the stages under mitosis. However, the nuclear membrane will disappear, the nucleolus will disappear, and the centromeres will start moving towards poles. So the characteristic of how the appearance of the cell would look like is very similar. Now notice under metaphase, you can recognize why is it metaphase, because you see the same equatorial pattern of the chromosome, that they are aligned right in the center, and the spindles are formed the same way. Kinetochores are used to attach them on these spindle fibers, or with the spindle fibers are actually made of microtubule. If you remember, we talked about microtubule. Let me get this right. I couldn't get a nice writing up here. So microtubule. Microtubule, if you remember, were structures that were giving a cytoskeleton to the cell. So we did mention the microtubules under the cell. So this is metaphase, very similar to the appearance of the metaphase under mitosis. But what's more important is to look at the chromosome and actually pay attention to the chromatids, because that helps you to look at or recognize the homologous chromosome. Now at anaphase, the cells, as you can see, what's happening is the tetrads are separating. So each end is getting half the chromosomal number. So it's separating. The homologous chromosomes separate and they move towards the pole. Now, very important note that anaphase 1, the cells are now haploid. It started off with a diploid cell, and now you have half the number of chromosomes. Now, anaphase is then followed by a telophase, which is very similar to the telophase properties that were taking place in mitosis. The same thing, you can see the nuclear membrane is forming back, and the nucleolus starts appearing. You also see the nuclear membrane forming, and you see the cells is actually dividing its cytoplasm and its content in half. And now at the end, you have interkinesis, in which the chromosomes still consist of two chromatids. Now at this stage, what has happened is the two haploid cells have formed. Now, in the next stages, we will see what happens to the fate of these haploid cells. So I will now conclude Meiosis I, part 1, and then we will look here Meiosis II in the next short mini lecture. So again, reviewing back, I'm going to go back. It's important to remember that the Meiosis I has these important features that take place, which is the reduction of chromosome due to crossing over and independent assortment, which means that the 4 chromatids are going to be separated out in 4 haploids, which we will see later on. The homologous chromosomes are separating. So far, we do not see the chromatids separated. So you still see, if you notice over here, you can still see the chromatids are still present in the same format. So you don't see the chromatids separating yet. So this concludes our mini lecture on this part, and then we will go on to the next section. Now let's look at meiosis part 2 and see as to what happens under meiosis part 2. Notice meiosis part 2 is written with your Roman numeral over here. You should be able to recognize meiosis 2 versus meiosis 1. In meiosis 1 we had only one Roman number, and this is meiosis 2. Now meiosis 2 begins with what happened just after meiosis 1. Now remember during the stage which is called as interkinesis, just like that interphase, but interphase was longer in the regular cell, and this is interkinesis because immediately the cell is going to go into a second kind of division. So there is no synthesis of additional DNA or duplication. That's why it's called as interkinesis. Now meiosis 2 is very similar to mitosis. So the stages of meiosis 2 is going to remind you of mitosis, and pretty much now, since its 2 sister chromatids are attached, the purpose of meiosis 2 is to separate out the 2 sister chromatids. Recall that mitosis was the same. We had 2 sister chromatids and they were separated. Now the difference is when the cell undergoes mitosis, it was a diploid cell. However, when the cell undergoes meiosis 2, it's a haploid cell. So here we have a haploid cell that we will see that it will separate out the 2 chromatids. So basically the same as mitosis, except cells are haploid and they're not diploid. Okay. So let's look at meiosis 2 from these images over here. You can see that in meiosis, actually, I'm trying to think. Did I skip a slide over here? Let's see. Let's go back. Look at the previous slide. No, we did do it. So what we wanted to point you is here you have the independent, the 2 haploid cells that were coming out, and this is the prophase. It's followed by a metaphase. Why is it metaphase? Again, you have the chromatids that are aligned in the center, which is followed by the telophase. I'm going to write that down. So the first stage is prophase, and this is going to be followed by metaphase, and after metaphase you will have the anaphase, and in anaphase you can see the 2 chromatids are now actually separating, and eventually, since it's going to separate out, and it will eventually give rise to what is called as the telophase. And in the telophase you can see that you eventually are having each nucleus having 1 chromatid within that. And notice that each of the chromatids is independently assorted. So you have one long red and one long red over here with the blue segment and one blue in here. So you really don't have two long ones on one nucleus, and you don't have two short ones on the other. So each of the two chromatids are totally separated. So you have exactly a short and a long chromosome in each nucleus. And eventually, after telophase, when the membrane will be reformed, the cell will eventually give rise to what are called as the daughter cells. And the daughter cells are now all haploid, and they have a variation of genetic material. They still have the two chromosomes that we began with, but if you notice, if you compare each of the chromosome, if you look at the long ones, this is a long red, and then you have a long red with a blue segment. We have a long blue segment and a blue with a red. So we have all kinds of 4 different combinations that is resulting. Now, if this variation did not take place, then the haploid cell will be identical to the parental cell. So this results in a unique variation in the genetic outlook of species. So keep in mind, Meiosis II had 2 parts, Meiosis I and Meiosis II. Meiosis II is very similar to mitosis. So this will end this mini lecture, and then we have one more short one to go after this.